Hi everyone, today we will talk about Illumina sequencing. I want to give you the tools or probably the language that you need to use to communicate with the core facilities. We will go through the basics, like what is a Illumina sequence and what are the basic components. Then we will talk a little bit about the length, different types of length and different types of mode. This is single or pair end, so we will revisit this concept. Then we will talk about the depth of the reads, how many reads you will get from the sequencing and how many you need. And we will introduce the concept of barcode to make the best use of the samples that you're going to use. Then we will talk about PCR artifacts and how to deal with that. So we'll introduce the concept of unique molecule identifier or UMI. So we'll wrap up with different types of runs, those including like single end, pair end, with barcodes, without barcodes, with UMI, different combinations, so that you have all the tools to specify exactly what you need with your sequencing facility. So let's start with the very basic concept of Illumina sequencing. So you will start with, there is here like a zoo of sequencing techniques, but at the end of the day, all of these will give you a piece of nucleic acid, DNA, to which you will have attached like these primers that are custom sequences designed by Illumina. And so this is the basic. You will have like these fragments with these primers, and then you will amplify these by PCR using specific primers that bind to this bit here and to that bit there, and you'll end up with this sequence flanked by these extended primers. These primers are typically the I5 and the I7. So you will hear about these sequences. And we will dive a little bit more in detail of how these primers can be changed. So this is the very basic and composition of your read. So you have your primers, and in the middle you have your insert. Now, let's move to the length of the different types of sequencing. So let's talk about like the length and the types of sequencing or modes of sequencing. So you will have your insert here, double-stranded DNA, and Illumina can only do DNA, and you will have your sequences corresponding to your primers in one end and the other end. And the idea here is that you will have the insert that can be of different lengths. Typically, you go from 20 to up to 1 kb, and we will see that these limits are mostly practical. So you don't want to have less than 20 base pairs because you wouldn't know where to map that read. And then you can go as far as 1 kb or probably even more, but that's rarely the case. You typically have it less than 1 kb. And the reason is that you have different types of lengths that typically you can have readings that are 35 base pairs or 70 base pairs. This changes with time, like there is the 110 base pair, 150, and there are some of the instruments now that go as far as 300 base pair, but not more than that. So a little bit this relationship between the length that you can have from the sequencing and your length will determine how we are going to read. So what you can do is two things. You can sequence from only one side. So you will start sequencing from here, and you can sequence part or the entire read. And what you will get is the sequence starting from here until you tell, like, I don't know, whatever limit this can be, I don't know, 35 base pairs, 70 base pairs, etc. Typically, this is done, for example, this single end read is when you can cover the whole length of your read with one go. So for example, if we have like this insert being 20 base pairs, you can run from this end like 350 base pairs, and then you will have all the information in this read. Sometimes it happens that you have a longer insert, probably, I don't know, 800 base pairs, for example, and you want to sequence as much as you can from the insert. So given the limitations of the length, if you do a read from 300, you will just reach to less than half of the read. The thing that you can do is to go and sequence from this other side, and you can do another 300. So you will have like 300 base pairs here and 300 base pairs here. And this modality is called paired end because you are reading the same read from the two sides, you will get FASTA files for one end and the other one you find for the other end, and then you will need to provide like the programs dealing with this type of read, like both files. In this case, so this is pair end and this is single end mode. In this case, you'll get only one FASTA file that corresponds to all the reads from this site. So that's the two modalities, single end, pair end, and that will depend on how much you want to read. If your insert is short, you are good with one single end. If your insert is long and you want to sequence as much as possible, you'll need to do longer reads and from both sides. Right, so now let's move to the depth. Right, so let's talk about sequence depth and barcodes. Each Illumina run can give you now like around 1 billion reads, and the question is how much do you need for your application? So this will depend the amount of read that you need per sample on the type of application. We tend to use more than what we really need. That is to say, once we reach a certain threshold, there is no more information that we gain. So probably if we sequence 50 million reads from one sample of RNA-seq, sequencing more than that is just wasting money because we'll not gain more information. So there are different papers depending on each application that tell you more or less how to do. I don't know what I remember, the last RNA-seq, after something like 20 million reads, the sequences is not longer informative. So we will go into the concept of lanes and flow cells in a second, but this means that in one run you could potentially put in the same run like at least a dozen, probably more samples, and you will have enough reads for each of them to be informative. So the question is now, how do you put samples together? So you use for this the barcode. So now I want to explain a little bit where these barcodes are typically incorporated, because they are put in somewhere weird place. Is not what one normally expect, but this is very Illumina dependent. For example, if we have our sample here, that we have like our I5 or I7 primer here, and we have our I5 primer here. Usually the first thing you add is like the 3' of these primers. So you come back and amplify, do the PCR with a longer primer, like around, I think there are 70 base pairs now, that have a sequence here that can be variable. So for example, and this is both for the I5 and the I7. So we have here a sequence where you can put the barcodes. So after the PCR, you end up with a situation where for one sample, you have like barcodes A, T, C, G. The barcodes can be like from 4 to 8 base pairs. You really don't need more because like you can put so many samples in a barcode, but more of that in a minute. So you end up with this read that has like the I5 and the I7, and you have this barcode incorporated here. If you want to add another sample, instead of using this primer with barcode 1, so the barcode is incorporated with the primer, you do a PCR with another barcode 2, let's call it, that will be a little bit different, but should be a little bit different or significantly different. And so you will go and do the PCR with a new primer, and you end up with barcode 2, that maybe barcode 2 is TC, TG. This is the reverse complementary. So that's barcode 2. And there you go. So here the barcodes are incorporated within this I5 or I7. And these are primers, these I7 or I5, of which you know what is the barcode. And you say, okay, I will use this barcode to label this sample. And you have to have this information. And you need to give the information to the core so that when the run is finished, they demultiplex or separate the reads depending on the barcode that they have. So that's the basics of barcoding. And I want to highlight where the barcodes are incorporated. And in the final part of the video, I will explain how that is actually recognized and sequenced, and what are the flexibilities that you have for that. So now let's move to the concept of UMIs and PCR artifact. Okay, so let's talk about PCR errors and UMIs, or PCR bias, let's call it bias, and UMIs. And UMIs is unique molecule identifier, or unique molecule identifier. Illumina is very dependent on PCR amplification, so there is a problem with that. So for example, we can start with a sample that has 3 reads, 2 are green and 1 is orange. Then we can have another sample that has 1 green read and 1 orange read. And if there is no bias, you will end up with double the amount of green reads and 1/3 of the amount of the orange reads. However, if these reads here amplify differently, so this amplifies better than this one, we can end up in the same situation. And this is not reflecting the original composition, but is reflecting the efficiency of amplification of these reads. For example, this read can have like, I don't know, secondary structure or CG-rich composition, and that skews its amplification. So how do we distinguish between the no bias and the PCR bias? So the idea here is to label each of the reads with a unique molecule identifier before starting the PCR reaction. So for example, going back to the same case, where we have 3 reads in one sample, and then we have 2 reads in the other sample. The idea would be to attach it, probably in one of the Illumina primers, before doing any amplification, one barcode that will be different for each of these violet molecules. And you can order oligos that have random sequences, and you typically use a code of 6 to 8 random sequences. But so if we have like random sequence X, Y, and Z in one case, and in this case we just have Y and Z, if we don't have PCR bias, we will end up with a situation where we have the same proportion of reads. But then if we go and look at the sequence of that unique molecule identifier, we will see that this has X and X, and this is different one. So these sequences here are coming from amplifications of the first original molecule, the same for here and the same for here. So then we can collapse all the reads with the same unique molecule identifier, and we go back to the original proportions. And in the case, so this is no PCR bias, so this is a little bit useless because we are collapsing everything proportionally. But in the case we do have PCR bias, we end up with a situation that we have 4 reads for the green, and all of them have the same unique molecule identifier, this YYYY, and then the set that is amplified less efficiently also has 1 unique molecule identifier. But then when we go and we collapse here, we only have 1 green read with 1 molecule identifier and 1 red or orange read with 1 molecule identifier. And here we recapitulate with what we have, and so we can manage that PCR amplification bias. So the important thing here is to attach these molecules with the unique modifier before each PCR amplification, which can be tricky, and probably the best way is to do this by ligating physically the 2 molecules. Sometimes you cannot do that. People do first round of cDNA, although that has some problems because there can be some lagging primer in the mix. Anyway, so let's bring everything together, and let's see the different types of runs that we can have. I want to introduce the concept of flow cells and lanes, because it will help you communicate and probably also understand a little bit the core facility, what they need and what their constraints are. So let's talk about runs, flow cells, and lanes. So imagine that we design our PCR amplification strategy so that we have the barcode on the I7, and this can be, I don't know, ATCG, and the reverse complementary. And then we incorporate the UMIs on the other side. And this is the same sort of concept. The UMIs are incorporated right in the middle of the read, and that will be important for what I'm going to explain in 1 second. So you have the I5 and you have the I7. So from this read that we have here, and this is our insert that is our molecule of interest. So you can have actually 4 types or 4 sub-reads from this read. So you can have like the first N coming from this side. You can have this one coming from the parent coming from this N. And then you have independent reads, mini reads, that will read you the barcode and will read you here the unique molecule identifier. Note that you can put barcodes on both sides or even unique molecular identifiers on both sides, or even you can swap where you put the barcode. So that's flexibility that you have, and you just need to tell, okay, I have my barcode or my unique molecule identifier within the I5 or I7 primer, and then also you will need to tell, depending on your read, you may want to do single end, pair end. If you do single end, you will need to tell, okay, sequence from the I7 primer or the I5 primer. So you do have all that flexibility. So if we label the runs like 1, 2, 3, and 4, you can have different combinations. For example, if you didn't put any UMI and you have barcoded samples of a short read, you can use like the read 1 and the barcode sample, and so you need this read here, and you can use any combination, and you can not put one or put the other. So that gives you a lot of flexibility, but here there is an issue that is very practical, and this is important so that you understand what the constraints of the sequencing facilities are. So each run is done on this called flow cells, and each flow cell has 2 or more lanes. So in each lane, you can put a mixture of independent samples. So you can run your barcoded mix of samples in one lane, and then another group doing something completely different will run in another lane their own barcoded mix of whatever they did. Those 2 reads will never like be in the same lane, and they will not be mixed, and they will be independent. However, there is a constraint, and essentially the problem is in most of the systems, you have to run both lanes using the same combination of reads, like either the pair end, single end, a given length from one side or from the other. So this can be tricky if you are trying to use a non-conventional reading. For example, you may want to sequence from this end 50 nucleotides and make an asymmetric run by reading like 250 from this lane. This is completely possible. The only problem is that you would need to use all of the lanes of the flow cell, because there will be no other group that will have the same requirements. So bear in mind what are the most common types of reads when you design, because either you will need to compromise and fit your reads to the needs of the most common user type. So that something to bear in mind is very practical, but sometimes it creates a lot of people. So with that, I think that's a wrap up. If you want to know about the next thing, the different types of bioinformatic file types, check this video somewhere that I generated. Very good. I'll see you next time.